We have, all right, so we are in the last part of the module 3, where we are talking about that how the mechanism, the molecular mechanism of how these vesicles are actually formed, find their destination, find their address, and the mechanism of a formation. So we talked about these slides, so I've just scooted a little bit back. So there are just only 3 types, COP1, COP2, and Clathrin. And these are the steps that from where they are going to, each COP2 goes from ER to cis Golgi, anterogate transport. This is cis Golgi to the ER, which is a retrograde backward transport. And Clathrin is everything else. Trans-Golgi to endosome, trans-Golgi to endosome, plasma membrane to endosome, Golgi to lysosome, melanosome, platelet vesicles. So these are all different areas that it can go. And of course, from plasma membrane, anything coming into the cell, that will be always Clathrin. These are the coat proteins and these associated GTPases, which actually help to anchor the coat proteins onto the membrane to pinch them off. So again, repeating. So these are all the SAR1 GTPases actually insert them onto the membrane of the place where the membrane vesicle needs to form. So these are the anchor, and on that anchor, these coat proteins actually hold on to them to pinch them off. And we already talked about the COP2 and the SAR1. This is the only one. Rest, all of them are actually ARFs. So we already talked that how the COP2 SAR1 happens. GTP associated SAR1 inserts its hydrophobic tail into the membrane. On that scaffold, all of the SEC1, SEC23-4, which are the COP2 proteins, associate, and the vesicle is formed. This is right here, the pinching of the vesicle. Once the vesicle is pinched off, the hydrolysis causes the coat proteins to assemble, and now it can go to its destination. So, talk about the destination in a second, but this is the second one that we are talking about. And here, this is the ARF, so all COP1 as well as the Clathrins, they use, all of them actually use this method. So here what you have, that there is a little bit different compared to the SAR1. SAR1, the entire part is a protein, and there's a hydrophobic tail that is tucked into the protein when it's bound to GDP. It extends that hydrophobic tail when it's bound to GDP or activated. Here, on the other hand, this is a lipoprotein. So a fatty acid is associated with the protein. So like lipid, lipo, plus protein. Unlike the SAR, which is a complete, just only protein. And what happens in here is that the GDP-bound form, the mechanism is exactly the same. The GDP-bound form has a structure where the fatty acid tail, which will not be stable in the aqueous environment of the cytosol, is tucked into a hydrophobic pocket. Now, this is your GNRP or GEF, guanine nucleotide releasing protein, or guanine nucleotide exchange factors, GEF or GNRP. These helps to exchange the inactive forms GDP with a GTP. Now, this is the active ARF. And see that now the hydrophobic tail of the fatty acid is extended due to the conformational change due to the GTP. This inserts itself onto the membrane. And then on this scaffold, this is the anchor, and now on this scaffold, the other coat proteins, which are the COP1 and the Clathrin protein that we have seen right here in this chart, right here. So all the COP1 codomer containing 7 different COP subunits, Clathrin AP1 complexes, AP2, AP3, depending upon where they are. These can actually assemble on there. And these are all your adapter proteins. Remember that those are specific as well. So we talked about that, how adaptive proteins actually do work. So this is essentially the mechanism. So this actually shows that how it happens. So you can see that these are the ARFs. On the codomer proteins, which could be COP1 or Clathrin, they all assemble and the vesicle will form. As soon as this vesicle is formed, what will happen? The GTPase activity with the help of GTPase, this GTP, which is bound in here with the codomer protein, will be hydrolyzed. So it will again become GDP, the phosphate, inorganic phosphate will be released, and as soon as it becomes inactivated, this tail will be retracted back to its original place. So this whole assembly will essentially be uncoated. So the coating is necessary just for pinching it off. Think about that. You just use your fingers to just make a small part of the balloon which is not properly inflated. You take it out and then you just move your fingers once that is done. I mean, like a similar mechanism, you got it. All right, so now what we got, that what's the mechanism by which vesicles are formed? We addressed that. What are the molecular signals on the vesicles that cause them to bind to the appropriate target membrane? This is the specificity that we also talked about that as well. So just make sure that you got this one because we already talked about these specific things. Remember that. Adaptins are the receptor-specific proteins that recruit the collaterin. We just gave you this example. So this is essentially, if you have a mannose-6-phosphate, there's a separate adaptin, whereas if you have a cargo molecule, which is going from the plasma membrane to the cytosol, it's a different adaptin. And these are based on the receptors that is in there on the membrane, because the receptor tails actually stick out of the membrane, and this is where they bind, and this is what drives the specificity. So again, just touching that, do not have to memorize, but see that right here, AP1, GGA, AP2, AP3, these are the adapter proteins which actually bind specifically to the receptor molecules. And here it has actually shown. See that right here, there are some specific regions of these coat proteins where they associate with these receptor molecules. So this is a cargo associated with the receptor in the vesicle, but this tail part sticks out, and that is recognized by a specific protein. And that is what we mean by the molecular signals on vesicles that cause them to bind to the appropriate target membrane. So this is what we did. And now we are going into how to transport the vesicle. So this is actually a Nobel Prize winning discovery that so these are called snare proteins, or in short, these are called the SNAP receptors. And there are also another class of proteins which are also present in them. They are called the GTPases. And this role is in the vesicle traffic and specificity and the fusion. So essentially, what is the mechanism of this? And know this very, very well, that what are the different space, and make your own notes. So what we essentially talked about beforehand, that how it is, so think about that this is essentially a mail being delivered from one city to another, and then delivered to your house address according to the address by a mailman in a mail delivery vehicle. So for each one of them, it is just not one component. So see that either the mail is delivered from one city to another by a truck or a plane or anything like that, goes to the post office. From there, it is sorted out. And then once the sorting is happening, it's going to a specific mail van, a delivery van. And from there, the mailman see the address, and then it is delivered onto your mailbox. So there are different components, and this actually works the same way as well. So the first part, we actually saw that how the vesicles actually form right here. That is what we're seeing, the donor organelle. This could be the ER, and this could be the Golgi, or this could be the Golgi transphase. This could be the plasma membrane, or this could be the endosome or lysosome. So this is your donor organelle and receptor target organelle. So here what we are looking at, so there are two mechanisms. The first mechanism is the mechanical force. Know this very well. Mechanical force for formation of the vesicle. This is just the pinching off the vesicle. And this is essentially your SAR1, ARFs, and then COP2, COP1, and your collaterals. These are the mechanical forces. Now, along with that, what you have, that specific things are packaged together, that is your adaptin. That those are the specific ones that is in there. So now you know that the vesicle is formed, and the codomer protein will get uncoated. So the mechanism is done. Now what happens, that how recognizes where it needs to go, there is an address that is also in this vesicle. This vesicle does have essentially a RAB GTP that we're going to see, a RAB GTPase, and the RAB GTPases do have, which is not shown in here, there is a cognate receptor for the RAB GTPases in the donor from the receptor organelle, and that confirms that there is the corresponding. So essentially, think about that you have your address written on the letter, and the mailman is going and seeing that your mailbox or your front door or the mail post does contain that number and the street. So that receptor will be that number on the street of your house, and here this is the letter which contains that address. Now, on the other hand, how do you know that you are packaging these proteins to go into this address? Remember that from one single post office, there are several mail delivery vans going out. This is a specific mail delivery van going to a single address. This is a different delivery vehicle going to a different part of the town. And this is essentially specified by the adaptants and the SAR1, SAR2, and the clathrin and the top ones. Remember that. That is basically packaging them in different destination places. Then finding the address. So now think about that. That suppose these are the one which are essential. This vesicle is going on the north part of the Clemson, from the Clemson post office. This is going to the south part of the Clemson. Now in the north part of the Clemson, you have the single addresses for each house. That is your RAB GTPases. And then, so what is your snare? The snare is essentially the mechanism by which the vesicle fuses mechanically with the receptor organelle. Which is, just think about that. This is basically the mailman hands extending out of the window, opening the mailbox and putting your mail and closing it. That will be your snare or the target snare. So notice very, very well that first you have the compartmentalization of the similar proteins going to the same destination. Then you have the mechanical pinching off of those. Then it is going by the RAB GTPase's address to the destination, and then the mechanical fusion of these two vesicles actually happen. So let's take a look at these. So right here, you can see a little bit more detailed view of these. See right here, you have a cargo, and then you have this stock part as such, and these are recognized by specific code proteins. We talked about that. And here you have the snare proteins, which are not working at this point, but these snare proteins will be helping in fusion of the membranes. And these are the GTPases like the SAR1 or the ARFs, which actually help in, they are anchored on the membrane, and these green parts are the code proteins, which are the mechanical force to pinch off the vesicle, essentially. So know this, that's what's happening here. So now, this is the coated vesicle budding from the donor organelle. Now it's uncoating, and the docking is assisted by the RABs that we are going to see, but that is not shown. We are going to see that. So essentially, there are RAB proteins on the surface of these, and there should be RAB receptor. We are going to see that in the next slide. RAB receptor, which is mailbox versus the on your mail. Now, this is the mechanical force, the target snare. This is present on the vesicle, and also you have a corresponding target snare in the receptive part of the vesicle where the target organelle is. And these will actually associate and pull this membrane down. Here it is sideways, the membrane is right here, and so it will be pulled sideways and it will fuse together, or it will pull down. So that's essentially what we are looking at. So let's take a look at a little bit of detail. How do the transport vesicles to their target organelle fuse, essentially, and this will tell you the perspective of the RAB GTPases as well. So here, what you are looking at, a transport vesicle, this is uncoated. So the code proteins are gone, SAR ARFs are gone. These are all with the same material that is going to the same destination. And now you have these VAMP. This is a specific name, and the V snare, remember that the V snare and the T snare, these are generic names. And what you are looking at is VAM, syntaxin, and then SNAP25. These are specific names of the proteins, which are some of the T snares and some of the V snares. So that's it. So it varies. Now, on the vesicle, you also have the RAB GTPase, which is bound with the RAB and is a GTP. And this is essentially the address. And in the target vesicle, you have the RAB effector, where the RAB GTPase will bind. So this is your letter and this address typed on your letter. And this RAB effector is a letter box with your street name or the house number or the street name, whatever it is. So that is the very important concept. Now what we are going to see that once the RAB GTPase and the RAB effector bind and find each other, they bring these two vesicles, this is the donor from the donor organelle, this is the vesicle, and this is the target, close together, almost like 10 nanometer or less. And then at that time, the V snare and T snare will interact, and there is an amazing molecular mechanism by which they will be pulled down and they will fuse, the membranes will fuse together. There's a supplementary video of that, so definitely take a look at that. So again, you do not need to memorize the individual name of these, but remember that the RAB GTPases are involved in the vesicle docking with the destination or the target. And there are more than 150 RABs identified, and each one of them basically is the destination for each. So if there is, so let's take a look at that and say how this is what's happening here. So if a vesicle is coated by a RAB1, then that is originated from the endoplasmic reticulum. And if it is going to the Golgi complex, the Golgi's cis face should have a RAB1 receptor. If it is going from the cis Golgi network, you can have a RAB2. If there is going to a secretory vesicles, they will have a RAB3A. Early endosomes will have a RAB4, RAB9 late endosomes. Depending upon where exactly it is targeted, you have these are the different addresses. You do not have to memorize those, but just know their role, that those are the addresses and the corresponding vesicle, the target vesicle should have the corresponding RAB receptor. That's the most important part. All right, so we got this. Now see that this is an amazing molecular mechanism that actually happens, which actually physically fuses the vesicle with the target. So here, the syntaxin and the SNAP25 and the VAM, all of them, the V snare and the T snare, they interact with each other and they actually form a coil-coil structure. It's a protein-protein interaction and a coil-coil structure. And as this coil-coil structure essentially forms, this vesicle is pulled down onto the membrane and it fuses. Remember that lipid will dissolve in the lipid. As soon as the phospholipids touch each other, they will just fuse by themselves. So that's essentially the mechanism. It's the snare complex, and this is essentially the way it looks like. This alpha helices that is formed with the VAM. VAM is the V snare, syntaxin and SNAP25 are the T snares, and they actually fuse together. We're going to see a video. I will show that you have that essentially. So now what happens? That you have this structure. So the membrane actually is brought down, and the membrane is fused with the vesicle is fused with the destination vesicle. Now what happens? That you have the V snare and the T snare. They are in this, which is we call as if they are in cis. Essentially, when they are separate, like they are from here and here, we call that the V snare and the T snare. They are in trans. On the other hand, when they are done with their function, right here, we call them they are in the cis formation, cis snare complex. And now they need to be recycled. They are not used up as such. So V snare, T snare goes back to their destination. And for that, there is a protein which is called the NSF. This is a hexamer right here, and I wrote down the name of it, which is not required. And along with that, you have an alpha SNAP, which is the snare attachment protein right here. And then, with the help of an ATP, they bind on top of that that structure, the cis snare complex, essentially, and separate them out. So this is with the help of an ATP. They disassemble the SNAP-25 syntaxine and the VAM, and see that right here the VAM came as from the vesicle that goes back to the destination assembly. So this is very important. So just know that what's the mechanism of essentially recycling back the snare complexes as well. So let's take a look. 1 second here. So let's look at. That I could just directly, this will be probably easy. So let's see. I will just I'll share that in a second. All right. Oh, sorry. So sorry. I'm just running like a little hamster on the wheel. Okay, all right. All right, I will just yeah. So I will just so this is a neurotransmitter fusion with the target. That's what is being shown. So it can be anything else. So, and why I'm not listening, hearing this? That's also funny. All right, so here I'll just narrate that right here. So after, yeah, so here, if you can see right here, this is a good picture of that. So the vesicle is, so the wraps are not shown in here. You have this picture in your supplementary videos. So the ones which are in the blues right here, these are your VAMs, which are the V snares. And right here, these are your T snares. There were syntaxin and the other ones, SNAP-25, and those are the ones. So as the vesicle comes in, which is not shown, that you will have the RAV GTPase is on top of that, and you have the RAV GTPase receptors on that. That will first engage, so that this vesicle and the target is close enough. When they're close enough, these V snares and the T snares can interact and see that how they essentially form that loop-like structure. That is essentially the process. So this is essentially the mechanism, and it's continuous fusion. So now you see that both the snare proteins are now in the alpha helical form like this, and this is your cis snare formation. When they were coming in, essentially, they were in trance, so they are in trance now, and once the fusion happened, they are in the cis form. Now you see the NSF and alpha snap comes in and essentially take them off. Essentially, this is your NSF and alpha snap by the help of this is the bottom one is alpha snap, the top one, and that with the help of your ATP, they are separated out, and now essentially they will just go back to recycle back essentially. So that's essentially okay. All right. That's good enough. All right, so let's go back to this. All right, so let's do some with like the some. All right, this is your first one. Or there's no trick. Just right. 1 minute is more than enough for this. The last thing that you saw in the animation as well. Good. Everyone? Stopping it? Last chance? Okay. All right. Yeah. That is correct. That is the assembly of the snare complex. Very straightforward. Just remember that sometimes we ask that this is with the help of an ATP. It's ATP driven. That's the only thing. I don't think that we need to go any more detail of that as well. So that one. Yeah. Anything else? No. Yeah. So that's pretty much it. Any question on this? Anyone? Anyone on this one? I mean, like, you may not have that today, but if you have any questions, just let me know, and let's go into the next module. So we will have plenty of time for this one to finish. So should not have any problem with those. All right, so now we are going into the cytoskeleton, and some of you actually looked at different microscope, like you have done some of the fluorescence microscopy in the lab as well, and you can see that some of these microscopes are confocal images. You have used the green, blue, red dyes as well, and the thing is that you visualize them separately, and then you can merge them, which you basically take a separate picture of each one of them, and you can see that. So what you are exactly seeing in here, just to so this is very straightforward. You can see the two spindle poles and the chromosomes essentially assembled in the metaphase plate during the cell division, and also along with that, you see the actins and the other ones in here. These are called the villi or villi. And these are not the microvilli which are present on each individual cell, but these are the finger-like projections. They are still tiny small, and this black space is basically the hollow or lumen of your small intestine, which is a jejunal ileum, the small intestine, absorptive and digestive part of your long intestine, the small intestine. So these finger-like projections are extended, and what you are seeing, the entire red border that you are seeing, those are essentially actin, and those areas you have microvilli, which are extending from each of the cells like finger-like projections. You cannot see this magnification. What you are seeing is a macroscopic structure right here. This is much larger. Not really macroscopic, but low magnification. But the red border that you are seeing, that is known as a brush border, which is the enterocyte individual cell with microvilli extending into the lumen, and it increases the absorptive surface area. So you see that these are called the crypts, which runs deep in here, and all of them are absorptive surfaces. So what you are seeing, the blue ones are the nuclei of each individual cells. these are the enterocytes. Remember that these are the cells that we saw sideways that takes up the glucose with the sodium synporter, carries it, and then to the blood side, it moves the glucose from the cell into the blood. So what you are seeing essentially, these are the entire red part, which are the actin, are the microvilli. These are the apical surface. On the other hand, the basal surface is with green, and there is a protein called laminin that is found only on the basal surface. And right here, this is where in the middle portion, this is where the blood vessels actually are extending, and the material comes from the apical surface into the basal surface into the blood. And here you can see some of the cross section of these blood capillaries in right here, and right here you can see that this part is a muscle, but you can see that there are some hollow structure in there, and those are the blood capillaries that are supplying in this middle part, and then they are moving from those areas. So this is what you are seeing in this picture. So now we are looking at these. So these are, some of them are very straightforward. So these are very straightforward. This is a cytoskeleton. You are looking at the different parts. And here, this is what the picture was looking like. So you are looking at this picture on the cover. So that red line is essentially the microvilli that is lined this entire finger-like projections. Remember that this entire part of this villi, which is a much bigger, is lined by those red. And what was stained is the apical domain, which was red in structure, and that is your essentially the actin. On the other hand, here on the bottom side, there was a protein which was called a laminin, not needed at all. That was stained as a green, so you know that it was on the basal surface of it. So this is essentially an epithelial cell which is fixed in one place. And if you look at that these microvilli are supported by this cytoskeletal structure, and along with that you have other structures. So what you're seeing, the red ones are the actins, then you have the blue ones, which are the intermediate filaments, and then the largest one are the green ones, which are the microtubules. So these three things, microfilaments, microtubules, and intermediate filaments, these are the three cytoskeletons. Microfilaments are made up of actin, microtubules are made up of tubulin, whereas the intermediate filaments, you have many proteins depending upon the cell that they are made up of with. But microfilaments are always actin, microtubules are always tubulin, no matter what type of cell they are. And this is a migrating cell. What do you mean by the migrating cell? This is a typical crawling cell, like your white blood cells. This could be a neutrophil, a macrophage, basophil, eosinophil. These are all motile cells that can move. They can move from the blood capillaries into the tissue space and look for pathogens, antigens, and they can move. And this is essentially the leading edge, which is the leading edge, and they form these phylopodiums or philopodia. These are feet-like structure which are formed by the help of the actin and help them essentially move. And the amoebas move the same way as well on the surface. These are essentially basically polymerizing these microtubules and depolymerizing the microtubules. So we already talked about this in detail as well. So the first, we are looking at the actin filaments, essentially what we are looking at. And these actin filaments, they are made of with actin monomers. And here you can see that they are 7 to 9 nanometer in width, and how each of the actins. So the blue one is one, the red, green, and the blue ones. Each one of them is one actin monomer, and they are associated to form a structure which is a filament-like structure. So if you find that individual actin, we call it is a globular actin or a G actin. So they are present in the cytosol. 1,000 million, millions of molecules of these. When there is a specific signal, they actually polymerize into this chain-like structure or filament-like structure, and there are two of them wrapped around each other. And this is known as the F actin or filamentous actin. So G actin and filamentous actin, they just go back and forth. So they assemble, disassemble, polymerize, depolymerize, same language. So remember that polymerization is making of the F-actin. Depolymerization is making of the G-actin from the F-actin. And it's a very dynamic nature. Now, each one of these filaments does have a plus end and a minus end. And we are going to take a look at what we mean by these plus and minus ends. All right, so first let's take a look at that where they're present. These are very self-explanatory. You should not spend much time in here. Know this from your blurb as well as from your recorded structure, microvilli, cell cortex, keep the cell shape. If the cells are attached to each other, these adherent cells actually stick them together. These are motile cells, formation of filopodia, lamellipodia. So filopodia are just finger-like projections, whereas a lamellipodia is like kind of a blunt structure. And also maintain the cell. Stress fibers is essentially runs alongside the cell, like in the direction where the cell is going, so that it doesn't tear up the cell as the leading edge is moving away. So that's essentially the important part. Then during the animal cell division, this contractile rim essentially formed. The ring is essentially that you know that the cells when they divide, they pinch off, and we call this as a cleavage furrow. And this cleavage furrow is essentially the pinching of the cells. This is also done by this actin or the microfilaments, essentially. And right, you have some couple of pictures in the EM. The next one, and we are going to talk about detail. We are just giving an introduction to each one of them. Then you have an intermediate filaments. You can see there are a little bit long. These are about 7 to 9 nanometer in diameter. These are about 10 nanometer in diameter. And where you find them is that they are found underneath the nuclear membrane and form the nuclear lamina. So this is actually essentially for the structural integrity of the nucleus, and they are found throughout the cell as well. Remember that for the intermediate filaments, this is a generic name. They have multiple proteins with all different names depending upon the cell. There is not a single name that what you have. And then the third one is a microtubule. This is a little bit different. Say the first one, microfilament, and the intermediate filament, the proteins are associated in such that they are not hollow. They are a solid structure, essentially, thin-like structure. Here, these are the tubulin monomers, and they associate with each other, and they form a ring-like structure. So the inside is actually hollow, and this structure is 25 nanometers. So you can see it's more than 1.5 times bigger than actin or intermediate filament. And they originate from a specific region in the cell, and we call this as a microtubule organizing center, or MTOC, which you know as commonly known as the centrosome. They radiate from those areas. Now, the microtubule organizing center could be also in different part of the cell, but they must come from one origin, which we call as an MTOC. MTOC doesn't need to be the centrosome only, it could be found in other areas, but that's from where they radiate out or grow and shrink. Unlike the intermediate filament or the microfilaments made up of actin, they can polymerize anywhere that they want. They do not need this organizing center to generate these microtubules like structure. So here is essentially a comparison with this. So if you look at the structure, see that the actins actually run lengthwise as such, the distribution. On the other hand, if you look at the microtubules and the intermediate filaments, what we mean by the similar distribution, that this is essentially the nucleus, that's empty part, this is the nucleus. And if you look at that, both of the intermediate filaments and the microtubules and intermediate filaments, they radiate outward from the central part of the cell. That's what we mean by a similar distribution. It radiates outward, this also radiates outward, but there is no such structural shape of the actin. They just run in all different directions in a cell. So that's why we say that they have the similar distribution. For which of the following has a similar distribution, and we give you the names. So choose that your microtubules, intermediate filaments have a similar distribution, which is radiating from the center of the cell towards the periphery, not observed in the microfilaments. All right, so your plus end and the minus end, and the structures that how uh, they are drawn essentially. Remember that the plus end is the fast growing, and we call this as a barbed end. And what we mean by barbed end is that they are a structure like this. So this is your plus structure, and the next molecule is on this, and this is your pointed end. This is your minus structure. So this is your barbed end, plus side, and the pointed end. We are going to see this in a second, but that is what we mean by this. And we are going to see that how they are associated with other things essentially. All right, so let's go into this. Now what we have, that when the actin actually polymerized into from G-actin, the first step that they need a nucleation from where the polymerization started, and then there is a rapid movement. So from G-actin to nucleation, this step is a rate limiting factor. Then it polymerizes really fast. But what happens that when you have the formation of the filamentous actin, there is a phenomenon that is observed which we call as a treadmilling, which means that the new monomers are added to the plus end, and the older ones are actually shed off from the minus end. And there is a state of equilibrium that they actually stay the steady state. So if the rate of addition and dissociation remains the same, the length of the actin actually doesn't change, but actually is a very dynamic structure as well. But if they're needed, depending upon where you need a dissociation, then the addition will be less, and it will really polymerize really fast, and it is also aided by some of the other proteins as well. So this is what is shown, the G-actin and the F-actin, they are in a dynamic equilibrium, and the G-actins does have something, this is a molecule, this is a chemical nature of that. They have a critical concentration. What we mean by the critical concentration of the globular actin, the critical concentration at that time in the plus end and the minus end are essentially the same, if the rate is the same. But what happens if the critical concentration is of the globular actin is greater of the globular actin is greater than the critical concentration, it will polymerize until it reaches the critical concentration. And if it is less than that, it will depolymerize. So again, I'm just repeating that, and will give you a much more clearer understanding. So all these globular actins, suppose these are there, and they do have a specific molarity at which they are in that cell. Say they are 3 micromolar is the concentration of the G-actin. And a filamentous actin will have a plus end critical concentration and a minus end critical concentration, which actually is determined by the cell, the nature of the cell and the nature of the actin filament. If the concentration of the G-actin, that 3 micromolar, if that is more than the critical concentration that is in here, suppose this is 2 micromolar, then there will be addition in here. On the other hand, say for example, if the G-actin, the 3 micromolar, is actually less than here, suppose here the critical concentration at the minus end is 6 micromolar, but I said that here the actin is present in 3 micromolar, so 3 is less than 6, so it would depolymerize from the end. So a plus end of the filamentous actin and the minus end of the filamentous actin do have these critical concentrations. So this is the important part. So this is what we actually see, and we'll look into. This is not the map, but this is just the idea that you have the globular actin, and then first it starts to nucleate, and then essentially it forms a rapid filament, essentially. So what essentially happened, that the nucleation is a rate limiting step, as I said. This formation from the G-actin to the nucleus actually takes some time. So if you're doing this experiment in a test tube, you see a lag, and then once the nucleation is formed, it rapidly actually increases the exponential state, and then it reaches the steady state. On the other hand, if you look at this graph right here, if you have added some preformed nuclei in the test tube, there is no lag. The curve is not sigmoid. It straight goes up into the exponential phase for polymerization and reach the steady state. So just remember these two graphs. We usually the graph and then ask you that what were the condition of the experiment, whether you have added nuclei or have not added the nuclei. So remember that if the nuclei are not added, you get a initial lag phase, which you don't if you add the nuclei. So that's really important. So this is what we meant by the barbed end and the pointed end. So here you can see the polymerization is happening. This is the barbed end, and this is the pointed end. And the actin monomers are charged with an ATP, and those are added to the plus end. Now, gradually, as we say, that there is a phenomenon that happens that they are gradually moved towards the minus end as new monomers are added to the plus end. And this phenomenon is called the trade mill. And this trade milling also does have another role. The actins do have an ATP step. So that means that it can hydrolyze the ATP to ADP, adenosine diphosphate, from tri to di 2. So if as these ATP bound actin monomers are treadmilling towards the minus end, gradually they hydrolyze it to the ADP. Now, ADP bound actin monomers don't have the same affinity to be bound onto the filament as the ATP bound. So the ADP bound actins actually come off much more easily. So now you know that why is the critical concentration are different. So the critical concentration in here, the ATP bound actin, is different than the ADP bound actin on this end. This is what we mean by the critical concentration, depending upon the concentration of the globular actin in the cell. So now what you can do that you can replace, you can replace these ATPs with an ATP analog, which looks like an ATP. It will bind it there, but it will not be hydrolyzed by the actin. So it is a non-hydrolyzable ATP analog. And these are used with many of these, we call this like nucleotide analogs in many of the drugs, antiviral drugs, essentially. Say, for example, remdesivir is an analog for that, which is incorporated into the formation of growing chain and cause chain termination because the enzyme cannot put anything on top of that. It's the same thing. Once it's bound, look like ATP, but it will not be able to hydrolyze by the actin. And now you have a stable structure. You have the plus end and the minus end. The critical concentration will be the same, and this will essentially not hydrolyze. And some of the poisons, essentially, which are cytotoxic poison, actually essentially does this. It stabilizes the actin, and it will cause the cell to die because there is no dynamic nature of the cell. It cannot perform its function, essentially. So here you can look at the plus and the minus end once again, and shows the treadmilling. Sometimes you can label specific globular actin with a fluorophores and see these, track these molecules. So these are the fluorophore tagged globular actin, and you can see that it can treadmill from the plus end towards the minus end, and see that the critical concentration right here, which is denoted as CC, is much greater than the CC plus. So the plus end, the critical concentration is right here, is low, whereas the critical concentration in this end, look at the coloring as essentially, is much higher. And that's the reason that the globular actins are added to the plus end, and they are depolymerized from the minus end. So know these concepts very well. So this will happen. And here you have one of the math related problems. So read the corresponding part of your textbook. That will help you a lot in here. So what we are looking at right here, you just look at the actin. This is your plus end. This is your minus end. This is ATP actin. And right here, see that the ADP PI actins, the PIs are going out. These are the areas where they are gradually being, the ATP is essentially hydrolyzed to ADP, and this is your ADP actin. Based on that, let's take a look at the critical concentration and how it is calculated. It's very easy. So what we are seeing that at this end, each of these globular actins are added at 12 micromolar per second. So this is the rate of association. The thick arrow is the rate of association, 12 micromolar per second. Whereas the dissociation, all reactions can be reversible. So it can be even with the ATP, although it is less chance, but there is always a chance that ATP-bound actin can also dissociate. The chance is less and is 1.4 per second. So that means that you can add 12 molecules of, essentially, if you just make apples to apples, essentially, without micromolar, you can add 12 molecules of G-actin, empirically saying, 12 molecules of G-actin to this end, whereas about 1.4 molecules will be dissociated to this end. That gives you the critical concentration, rate of dissociation over rate of association. So it is 1.4 over 12. And if you calculate, it will come out like a 0.116 or something like that. And that is 0.12 micromolar is the critical concentration at this end. So definitely remember this formula. It's D over A. Dissociation over association. That's it. Very simple. And we can give you just like a whole number so that it's easy to calculate. On the other hand, this is the minus end. Look at this right here. The association dropped tenfold, 1.3 micromolar from 12. It's 12 micromolar. Here the rate of association is less, and the rate of dissociation is 0.8. It is still less, but see that what is happening here, the chances of ATP actin being added is drastically less from this, and that gives you 0.8 over 1.3. It gives you about 0.6 micromolar is the critical concentration in this end. Now the moment of, this is the moment. So remember that this is what I'm saying. Make sure that you get that. Suppose in the cell that you have 0.3 micromolar of G-actin available in the cell. 0.3 micromolar. So if you think about the 0.3 micromolar, 0.12 micromolar is less or more than that. 0.3 and 0.12. So what will happen here? Let's go back, right? Oh, sorry. Let's go to this right here. You go to this formula. If your globular actin concentration is less than the critical concentration, it will depolymerize. If it is more than the critical concentration, it will polymerize into the F-actin. So you know this formula. This is the truth. And right here is your values. So 0.3, I give you the value 0.3. 0.12 is less than that. So you have a net addition at this end, whereas a 0.3 is 0.3. I'm sorry, 0.3 is more than this, or this critical concentration is less, whereas a 0.3 is less than this, or the critical concentration is more at this end. So that's why you will have dissociation. Make sure that you get that. So the number is in between that is given. On the other hand, say for an example, if you give a concentration of, say, you have 1 millimolar of actin present in here, 1 millimolar, and that's where things can get complicated. If I put a value in between these two, in one end it will be less, one end it will be more, so that's no problem in calculating. But if I just put 1 millimolar, you can see that this is also less than 1 millimolar, this is also less than 1 millimolar. Still in that condition, this end will add actins and this will dissociate actin. And this is where people get confused. Remember that the reason is that from 1 millimolar, this value is smaller in number compared to this. So they are both smaller, but this is more smaller. Does that make sense? Well, there's nothing said that it's more smaller, but I'm just trying to make it up. But it is 1 millimolar. This is closer to 1 millimolar. This is much more distant from the 1 millimolar. So this value is even smaller than that. So even if the value of that is more than both, wherever is the smaller value, you will have addition of globular actin to that end compared to the other. So just make sure that you remember that these two scenarios where the value could be in between, and that is easy. But if the value is more than both, still the one with the less the critical concentration will have polymerization compared to the other end. So that is very, very important as well. So let's take a look at this. So let's do this math problem. Just use your formula, rate of dissociation D over rate of association D, and then calculate that which is bigger, which is smaller. Minutes. Right. Don't rush. Take the time if you just understand for a moment. One. Good. All right, stopping it. Yep, that is the correct answer. So now let's see that what happened is I talked about two conditions in between. So condition 1 and condition 2. So this corresponds to which one of them? 1 or 2? Anyone? Previously I said the value in between and value more than both of them. Yeah, so 2. Yeah, that's correct. So see that what is happening right here. Just calculate the D by A, D over A for each one of them. Dissociation is 2 over 10.2. Everybody good? And then D over A 2.55. This is 0.5. So that 0.2, 0.5, both of them are less than 2.5. And that's what I was trying to say. But what is happening, that 0.2 is less than 0.5. So this is the word I was using. This is more smaller compared to this. And that's the reason that you will have a net addition to this end compared to that. So whenever you calculate that, if it's, say, for example, 0.2 and this is 0.5, if I give you the value of, say, 0.3, super easy. 0.3 is more than this, so it will have addition. 0.3 is less than this, so you have a dissociation. There's no problem. But if the value just like this is more than both, then the smaller critical concentration value will have addition compared to the other end. It still will happen, actually. So this is essentially you interpret the data with. Good, everyone, with that? So you may see a simple math problem like this in your exam. You will not need a calculator even. Just remember that E over A, that's it. All right, so some of them you have used in your cell biology laboratory. If you're not taking it, that's why I just same. So, or you have not used yet. So you will be using the cytochelacin. This is a fungal metabolite. There are many of them. Cytochelacin A, B, D. There are many different ones. They are very close together. So essentially what they do, the cytochelacin, they will bind and depolymerize the actin. So it is a cytotoxin. On the other hand, there is also some of the toxic sponge. Latrine is a compound that will also cause the depolymerization. Now see that we already talked about this stain, and you have used in the lab the same stain. This is phalloidin, and where you get this from? This is from the death cap mushroom. This is very common in South Carolina. If you know that, you will be able to see that usually it does have a kind of a bulbous structure that from where you can distinguish that death cap mushroom. It's everywhere. And that's Amanita phalloides, and from where you can get this phalloidin. See, there is a difference in between the function of that. See, the cytochelacin latrine, they depolymerize the actin, whereas this actually stabilizes the actin. It doesn't allow it to depolymerize. It binds on the side of the F actin and stabilize them, and that is taken advantage in cell biology study to stain. So you have a dye or a fluorescent dye associated with a phalloidin, and it goes into the cell. Once it's fixed, it binds on the side of the actin, and essentially your actin actually fluoresces. So this is your phalloidin. Essentially, these are some of the tools in the cell biology to understand many different phenomena. This is your effect of cytochelacin of a leading edge of a cell. So if you carefully look at that, this is your phase contrast. Remember that. So we are looking at, and this is the reason that where we need a living cell to see these two different ones. See that this is your Lomarsky right here, DIC. You can see that there are these tiny projections or these fiber-like projections. Those are your actins. Whereas you can see, if you see carefully, right here there is an edge where you don't see any actin. The actin is retracted from the plasma membrane to this, and this is essentially the effect of cytochelacin. So these two are live images, but just to make sure that you get the same thing, after this step, the cell is actually fixed, permeabilized, and then you did the immunofluorescence microscopy, and it shows that this is the leading edge, which is kind of right here. So you can see that this gives you an idea, but the contrast is not enough. But you cannot see these phenomena like you will be able to see this, but in the same cell you will not be able to see the next step unless you use a low contrast Lomarsky or a phase contrast essentially. So this is one of the applications that where Lomarsky or phase contrast, even with their low contrast, actually come in play, because immunofluorescence is going to kill the cell. You can see the final effect with that, but in the same cell you will not be able to see this and this in the same cell by immunofluorescence. So that's essentially the effect of cytochelacin. You have done with actin, green fluorescent protein with phalloidin, and the nucleus is stained with a blue stain, which is called a DAPI. Essentially, that you will use in the lab as well. All right, so after that, we are looking at some of the actin binding proteins. So these are essentially some memorization type of things. So just if you can just make some cards or those and look at these every day, because there are lots of names in the second unit 4. And if you see them every day, it will go into your long-term memory, essentially. So profilin is a positive regulator and thymosin beta is a negative regulator of polymerization. We're going to see that. Profilin, gelsolin, ARP2/3 filament, we're going to see that. And then we're going to see the motor protein stability, CAP-Z and tropomodulin and tebulin sometimes later. So let's take a look at the first ones, essentially. So here, what we are looking at the top is actin filament, plus N minus N. And I'm starting from the minus N. So the actin ADP, you know that the actin ADP actually will dissociate depending upon the critical concentration. But the rate of dissociation for practical physiological purposes are slow. When a cell is actually moving, you are polymerizing, depolymerizing these actin, and that needs to happen in microsecond speed. Like, they are extremely fast. And the natural dissociation of actin ADP molecules one at a time is not enough fast. So there are some process to speed up the process. Cophilin is one of the one. This actually severs chunks of these actin ADPs and then recycle these essentially. So cophilin, again, remember that these are severing proteins. They just cut off the tail of the actin super fast. So now the monomer is actin ADP. So D means this is a globular actin associated with ADP. And what we are going to see that this actin ADP is essentially, so this is your cophilin. Cophilin is bound onto the side and helps it to sever, and it will be recycled back. So this actin ADP is now bound to another protein which is called the profilin. Now remember that we just went to the previous one. Profilin is a positive regulator for polymerization. What it means that once the profilin binds to the ADP, it changes its conformation so that it can replace the ADP with an ATP. So this is like your GNRF or GNRP, the protein that is helping to replace the GDP from GDP. Here, the profilin is helping the movement of ADP out and recharge it with an ATP. Same kind of function. When it is ATP bound, that molecule can bind to the plus N. So the ADP charged molecule is now charged with an ADP. It can be added to the plus N. Now, there is a negative regulation as well. So know this very well. This is where a lot of people get confused. Thymosin beta 4, it's a negative regulator of the addition of the actins in this plus N. So what is happening? That suppose a cell was actually moving towards a target, and at that time you need to polymerize this rapidly, and this cycle, the cophilin-profilin cycle, is going on. It's extending on this way, and it is basically severing or depolymerizing from this end. It is growing, and the cell is moving. And then it reached its destination. It doesn't want to move anymore. So at that point, what will happen? That all these actins that are depolymerized, if they are charged with an ATP, they will still tend to bind it here. So how the cell actually controls that process? They process it with this thymosin beta 4. So upon signal, the cell doesn't need polymerization. This thymosin beta 4 will sequester the ATP actins. So what it means that, think about that, you have the pool of ATP actin in the cell, and it will sequester that, so this ATP actins are not available to go to this end, and that will essentially stop the filamentation, the process. So let's think it in a micromolar way. So we already talked about that. The critical concentration here is actually less, here it is more, and you have a critical concentration which is more than this end, so it is being added. Now, what is happening, that thymosin beta 4 is taking away a few thousand or million molecules of the ATP actin and taking it out of the pool, so they are not available, so the concentration of ATP is decreasing. Suppose I'm just saying that this is a 2 micromolar is the critical concentration, and initially it was 3 micromolar, and it will have polymerization. And due to the thymosin beta 4 sequestering it dropped to 1 micromolar. So 2 micromolar is more than 1 micromolar, so the polymerization would stop. Basically, it reduced the available ATP actins in the pool of the cell to essentially compensate the polymerization. So know this cycle very well. This is an important concept as well, that how this cophilin, profilin, and the thymosin beta 4 control the polymerization regulate the filament formation essentially. All right, so after that we are looking at the actin polymerization. This is a very straightforward process. This is a motile cell like a white blood cells, like a neutrophil, macrophage, other cells moving on a substrate, which is a tissue space, and each place that is attaching with the substrate is known as a focal adhesion right here. And in the step 0, this is at step 0, the trailing edge must adhere to the substrate before the lamellipodium can be extended, which is not shown. So here, right here, see that lamellipodium is extended. The focal adhesion must essentially be, see that this is your extension number, this is 1, so this is what I take as a 0. So you have the attachment, lamellipodia extending right here, and then there you have a new adhesion. And the width of the cell body, and this happens totally due to the actin polymerization towards this end. And then finally, the old adhesion, this will come off and the cell will basically move forward. So know the extension, adhesion, translocation, and the deadhesion, these phases. Sometimes you may get a picture and you may have to tell that what is going on in here. And so this is essentially looks like a fried egg, but this is basically the leading direction of the migration. And these are the zone of polymerization, the ones which is essentially being shown in these areas as well. So yeah, I mean, like this will take a little bit of more time, so we will not start that. We will start with the ARP23 complex with that. So I will open up the module 2 practice quizzes as well. It may not make sense much, but still, if you find a couple of basic questions on dissociation, association, profiling, cofilling, you can answer those. All right.